Okay, so this is more of Chapter 6, Acellular Pathogens. We have to go over a couple new ones, Prions and Viroids and Virusoids. Um, Probably the main one is the Prions, and these are other things that are not alive and kind of weird. And so, oh, I should show you that. That's a picture of a mad cow. And so this is, we're going to talk about mad cow disease or bovine spongiform encephalitis, BSE. So last time we talked about viruses. They're not alive, they're acellular, obligatory intracellular parasites. Um, they have a genome, they have a protein coat, they infect cells, very, very small, unusual. Okay, virus structure includes always a nucleic acid, but it could be DNA or RNA, never both. Always a capsid, then maybe an envelope, maybe some spikes, maybe some enzymes. It varies. All viruses, though, have to get inside of cells, and then what happens depends upon the type of genome they have and the kind of enzymes they have. All viruses attach, penetrate, get their nucleic acid uncoated. They might insert their nucleic acid into the chromosome, but to replicate, they then use host organelles, host enzymes, host resources, copy their materials, assemble, and leave. What's unusual, or there's many unusual things about viruses. Among those is that they can cause cancer. They have genes that can take over the cell and cause the cell to begin not responding to normal controls on cell growth, cell location, cell activities. Okay, so that's kind of a review, but now we're going to deal with some unusual acellular pathogens. The first one are the viroids. These are circular RNAs, which generally don't occur in many places. They're, there's no protein coat, there's no envelope, there's no enzymes. They replicate, they get the host cell to replicate them. Fortunately, we don't know of any of these in people, but they're common plant diseases. They include a bunch there: the potato spindle tuber virus, the tomato plant matcha viroid, avocado sunblotch viroid, and peach latent mosaic viruses. As you can see, they affect crop plants. There's more of them. This is just the ones that become important. I don't have a diagram of these guys. But they do weird things. We think that they're normally spread by insects feeding on plant parts, and in cultivated plants they can be spread by machinery that injures the plant a little bit and then gets new viroid on the plant. We do have something similar in humans. These are virusoids. These are small, self-replicating, and when I say self-replicating, that means inside the host cell. They're circular, they're single-stranded RNA. They produce very few or no proteins, and they require a helper virus. Sometimes they're referred to as defective viruses. The best example is the hepatitis delta virus, or hepatitis D, which is never found on its own in a patient, is always with hepatitis B. And so it's a co-infection. It's another, naked, no proteins involved. So they kind of think, well, it's a defective virus that just lost its protein coat and commonly occurred with some other virus. Okay, but it gets weirder. We've known a long time of a couple diseases that we couldn't identify the organisms. These really came to the forefront in the 1990s. It turns out these are all protein-only diseases. These diseases replicate by converting a normal cell protein to a disease form or a prion form. And that prion form, once it's been converted, that prion form will convert other normal proteins. And this happens in nerve cells in the brain, and so it damages brain tissue. It causes accumulations of proteins that kill cells, and then the brain has areas of dead spots, and it starts to look spongy. So these are called spongiform encephalopathies. And so there was a big outbreak of these in the 1990s and 2000s. And so you kind of see in England where this happened, and I'll give you more information on this. But it was recognized in cows that this was spreading, and eventually some of these were found in people. So prions it stands for proteinaceous infectious particle. They can be inherited. They can be transmitted by ingestion, transplant, and surgical instruments. There's a whole bunch of these spongiform encephalopathies. The one we've known about longest is a sheep disease called scrapie, but we've known about certain families in Europe that have shown this, and so it's sometimes called Creutzfeldt-Jakob disease or Gertzmann-Straussler-Schenker disease, or sometimes fatal familial insomnia. We know of this from, I think it's Papua New Guinea. There it's called kuru. In cows, it's called mad cow disease, or now we have it in white-tailed deer, where we call it chronic wasting. So what's going on here is that there's a normal protein that's produced by host cells and is found at the surface of the cell. If that comes in contact with the disease form, here shown as this PRPSC, the abnormal form will start converting the normal form into more abnormal forms. And this just keeps converting and converting, and this stuff accumulates. And as it accumulates, you get this conversion, and you start to get areas of dying cells, and so it looks spongy. The disease form of the protein is really tough. It withstands acid digestion. It lasts in the soil. It can withstand processing, and if it's ingested, it'll make its way from the intestine to the brain. So it's hard to get rid of. So it's basically a misfolded protein, and that misfolded protein just seems to last for a long, long time, and it produces motor abnormalities that show up early, changes in how people walk and move, tremors, and eventually this will progress to dementia, maybe insomnia and then dementia. In mad cow disease, this turns out to have been spread by a processing of downer cattle. And they would process these cattle that couldn't be sold for human consumption. And they'd process them into a supplement for young cows. And it turned out this spread mad cow disease in herds in England. And so the number of cases skyrocketed in the early 90s. And eventually this led to a dramatic operation to remove cattle from England. And so all the cattle in England were eventually slaughtered because of the rapid spread of this. Initially, agricultural people in England tried to say this was never going to affect people, but eventually cases started accumulating. And so we started to see cases in people. It eventually showed up in North America, but never to the extent that it did in Europe. In Europe, they wound up having to slaughter 4.4 million cows in an effort to contain the disease. And despite efforts to assure the public that it wasn't going to affect the public. We now know of at least 178 deaths that occurred in the public from ingesting beef that was contaminated with mad cow disease. So pretty dramatic. Um, and we have some additional types of non-living acellular pathogens, viroids and virusoids. Those have a genome but lack any protein. Prions themselves consist of misfolded proteins and nothing else. There is no genome associated. These have various replication cycles. We don't call it reproduction, but it's replication because these are really chemicals. Um, and so some of these are causing plant and human disease and animal diseases.